will discuss about gram-negative bacteria today. A bacteria can be differentiated into two types based on the gram staining, that is, a gram-positive bacteria and a gram-negative bacteria. A gram-positive bacteria takes up a purple color, whereas a gram-negative bacteria takes a pink or a red color. This difference is because of the stains they take up. A gram-positive bacteria takes up the crystal violet stain, whereas a gram-negative bacteria takes up the color of a saffron. And this difference is because in a gram-negative bacteria, after which a crystal violet is stained, in the second step it is washed with an alcohol. So, in this step, the crystal violet is completely lost, and these bacteria take up the counter stain, which is the saffron. This difference is because of the cell wall composition. A gram-positive bacteria has a thick peptidoglycan layer, whereas a gram-negative bacteria has a thin peptidoglycan layer. So, in the absence of a thick peptidoglycan layer, it is less strong to hold the primary stain. So, the primary stain is easily washed off in a gram-negative bacteria. A gram-positive bacteria takes up a purple color, whereas a gram-negative bacteria takes up a red or a pink color. The examples of gram-negative bacteria are Escherichia coli, Chlamydia, etc. This diagram shows the different gram-negative bacteria, that is, an examples of different gram-negative bacteria. Next, we will discuss about the characterization of a gram-negative bacteria. So, the different shapes or in which a gram-negative bacteria are found is from a rod to bacillus, from a bacillus to cocci to a spiral shape. So, in different shapes, a gram-negative bacteria is found, and these shapes vary. However, some shapes may be cocci bacillus, tetrads, and trichomes. These are the different shapes in which a gram-negative bacteria are found. First is the diplococci. A diplococci is usually an aerobic bacteria. A cocci bacilli is also found. A bacilli is a lactose fermenting bacteria. A comma-shaped. These are oxidase positive bacteria, and they can be classified into three types, that is, which grows in 42 degrees Celsius, and another category is which grows in alkaline medium. Next category is the bacteria that can produce urease. This diagram shows the different shapes in which a gram-negative bacteria are found. So now let us discuss the gram-negative bacterial cell wall. The cell wall of a gram-negative bacteria is more complex than a gram-positive bacteria. That is, even though this peptidoglycan layer is very thin, there has a more complex structure compared to the gram-positive bacteria. So, in a gram-negative bacteria, it has a thin peptidoglycan layer, whereas it has an outer membrane, and this outer membrane is absent in a gram-positive bacteria. So, in a gram-negative bacteria, it has a thin peptidoglycan layer as well as an outer membrane. And this outer membrane is thick, that is, it has 7 to 8 nanometer thickness, whereas the peptidoglycan layer is very thin, that is, it has only a 2 to 7 nanometer in thickness. So, there is another space between the cell membrane and the cell wall, and this is called the periplasmic space, made up of periplasm. So, this layer is also seen in a gram-positive bacteria, whereas in a gram-negative bacteria, this is much larger than a gram-positive bacteria. The peptidoglycan layer in a gram-negative bacteria is thin, and this layer is found above the plasma membrane, and this occupies 5% of the cell dry weight. And in some bacteria like E. coli, it has 2 nanometer thickness of peptidoglycan. So the peptidoglycan in E. coli is 2 nanometer. So now we will discuss the components of a gram-negative cell wall. So first is a periplasmic space. A periplasmic space in a gram-negative bacteria is made up of several proteins. So the periplasmic space is made up of several proteins, and this assists in acquiring nutrients such as hydrolytic enzymes, and that they attack the nucleic acids and phosphorylated molecules. It is also made up of binding proteins. This helps in the transportation of several materials into the bacterial cells. So they also have certain enzymes which synthesizes the peptidoglycan layer and modify the toxic elements that cause harm to the cell. So the periplasmic layer is thick than in a gram-positive bacteria. So this layer is made up of several proteins including binding proteins. So next we will discuss about the outer membrane. The outer membrane is seen above the peptidoglycan layer. So outer membrane is above the peptidoglycan layer, and this layer is made up of brown lipoprotein. So this layer, that is the outer layer, is made up of brown lipoprotein, and this is covalently bound with the peptidoglycan layer. So the brown lipoprotein is covalently bound with the peptidoglycan layer. The gram-negative cell wall also has the adhesion sites, that is, these sites are present at the outer membrane. So these adhesion sites allow the cell contact as well as the membrane fusion. That is, through these adhesion sites, the substances are allowed to enter to the cell. So the outer polysaccharide is made up of 2 layers, that is 2 leaflets, and the outer leaflet consists of lipopolysaccharides, and this lipopolysaccharides consist of lipid A, core polysaccharide, and O antigen. So the outer leaflet consists of 3 components, that is lipid A, core polysaccharide, and O antigen, and the inner leaflet consists of phospholipids. So the outer membrane consists of 2 parts, that is 2 leaflets. The outer leaflets consisting of lipopolysaccharides and the inner leaflet consisting of phospholipids. So the outer membrane has porine proteins, that is, these proteins help in the transport, that is, in the entry of small molecules like glucose. The larger molecules like vitamin B12 are transported across the membrane, that is, across the outer membrane by specific carriers. This also prevents the loss of components, that is, the outer membrane prevents the loss of components, especially from the periplasmic space. In a gram-negative bacteria, the teichoic acids are absent. A gram-negative cell wall is shown in this diagram. A gram-negative bacteria has an additional membrane apart from the cytoplasmic membrane. This is the outer membrane. A gram-negative bacteria has a very thin peptidoglycan layer, whereas the periplasmic space is larger than a gram-positive bacteria. The outer layer has porine proteins within them. That is, it is these proteins that help in the entry of small molecules into the bacteria. The outer membrane also consists of lipopolysaccharides, and S layer is being attached to the outer layer. The S layer is a part of the cell envelope, and this layer is composed of proteins or glycoproteins. I hope the concept of gram-negative bacteria is clear. For more such videos, do subscribe to my channel. If you like the video, please like as well as share with your friends.